biological microfluidics lab. This is where we perform microfluidic device fabrication. Everything starts off with the mask design phase, where we use CADing software in order to actually fabricate the designs that we want to do. So this is one example. This is the mask. It's on a PT or a mylar sheet. You can have them on a chrome sheet as well. So you have the different designs that you want to fabricate, and this is the end result. I'm just going to touch it with my hands because it's an old device and I'm not going to use it again. So you can see the structures kind of. Okay. So this mask becomes this microfluidic device. What we usually do is, after we have our masks printed, we have to prepare our silicone wafers. Yeah. So you do master wafer preparation, and we can either use silicone wafers or we can also use glass slides. So it's either this one or this one. So we either have our devices here or here. What we do is, let's say for example we're using the silicone wafer. First of all, we start with the spin coater. So of course we prepare our wafer by performing piranha treatment and other types of treatments. And this, okay, there. So we use really deposit a UV photoresist. So it's kind of like a resin that's UV sensitive. We use SU8. It's called SU8 photolithography, and we use different numbers based on the thicknesses that we want. So this just indicates the viscosity of our photoresist. What we do is we deposit this on our spin coater, we activate the vacuum, and then we pour our photoresist on top. We choose the program that we want, aka the spinning speed, depending on the thickness of the photoresist that we're after. After we finish with the spin coating, we perform the soft baking. So soft baking is composed of two steps. Baking the wafer on a 65 degrees Celsius hot plate, and then a 95 degrees Celsius hot plate. After which we expose our silicone wafer to UV, and we align our mask. This was built here, so we align our mask on top of our silicone wafer, and then we expose it to UV. After we expose it to UV, the hollow areas allow UV to go in. So our SU8 resin, it cross-links, and after it cross-links, we perform post-exposure bake. So you also put it on 65 degrees Celsius and 95 degrees Celsius, and the timings depend on the thicknesses that you're after. Afterwards, you perform SU8 development. So this allows all the uncross-linked polymer to wash off the surface. After that, you have this silicone wafer. So this is the finished product. Okay, so this is the mask that's been imprinted on your silicone wafer. After that, you prepare a solution of PDMS and you pour it on top. Once you pour it on top, you put it in the vacuum. You can go see the vacuum. So you put it here. Right now we have one device that's in vacuum. So this removes all the air bubbles from the PDMS solution. Once all the air bubbles are removed, you place them in the oven in order to curate it. So we have this for the PDMS to dry and to set. So once it sets, you can cut off the device that you want. Once you cut it off, you put it into individual microfluidic devices. You use glass slides and then a microfluidic device. You place them in this plasma treatment. The plasma treatment renders the surfaces hydrophilic, so once they're hydrophilic, they can stick together. Once they stick together, you can puncture holes for the inlets and the outlets. So we have different pens depending on the size of the hole that we want to puncture. So once you puncture your holes, you can put the tubes in order to have your fluid circulate inside the channels.
What are your microfluidic devices used for?
So it depends. For us, this lab is a biological microfluidics lab, so we all use it for different purposes. We have people who use them for bacterial motility, for bacterial computation, DNA computation. I use them to stimulate blood vessels to see what happens inside blood vessels. Yeah, so basically you can use them to mimic human organs or to understand the behavior of microorganisms. So yeah, we also have a Venice maze. So
Pardon?
Venice maze. So
Okay.
like the actual roadmap of Venice. What else? Just to understand traffic and to kind of find solutions for traffic. Each one of us can talk to you about their projects a bit better, of course.
Yeah.
But yeah, this gives you kind of like an overall idea of the kind of projects that we have.
What we see here is a confocal microscope. There is a series of steps in which we operate this to turn it on. So first, what we have here is a light source. So we turn the light source on. Just be careful because they have optical fibers here. So once the light source is on, we have controllers. We turn that on and it turns the camera on. This is a confocal microscope with this spinning unit. It's more essential and critical because most of our experiments are not just imaging but also video recording. So to take time-lapse images and take a lot of frames at a very short duration and acquire more information. This is really helpful, especially with the spinning disc unit. So this is where we operate. There is the objectives, and this is an inverted microscope. So the objectives are from the bottom, and imaging is done from the top. This is a stage where we place our samples in. For example, I have a microfluidic device here. What we do is we place our sample in the holder, secure it, and we place it on the stage to image it. We have a specific software that we use for imaging, which is Metamorph Advanced. So with this software, we can obtain live images, live videos, record the videos, and also set time-lapse imaging in a way like for a certain number of hours and seconds. And another advantage is that we can do Z-stack imaging, that is not just X and Y, we can also acquire information on the Z plane. The most important reason why we use it is for biocomputation experiments and also experiments with bacteria and other microbes. This has 3 filters for fluorescent bacteria. One is M-cherry, which is red, GFP, which is green, and BFP for blue fluorescent bacteria. I have an image here of red fluorescent bacteria. You can see that the bacteria, these are the bacterial cells at 4x objective, which is genetically modified to express M-cherry fluorescent protein. So it fluoresces red. This way we simultaneously can image 2 or 3 colored bacteria and see how they differ or their motility pattern is or their behavior in the device itself is. So what we see here is euglena cells, which is kind of running through or navigating through these microfluidic channels. You see that this is a short video or it is a time-lapse video, but because of the disc spinning unit, this video is set in such a way that we can capture about 5 to 10 frames per second. So there's a lot of information that we can gather instead of just losing even per second. So that is the biggest advantage of using microscopy like this.